Hi, everyone. I'm really happy to be here and excited to share a little bit about our ongoing work using plasma proteomic signatures to capture unique cardiovascular risk phenotypes among people with and without HIV. So people with HIV experience higher risk and earlier onset of myocardial disease compared to people without HIV, and that manifests in several different ways, including clinical heart failure. So a nice example here on the right from VAX are some estimates of association showing higher hazard with seropositivity that's markedly higher among younger persons. We also see higher risk of subclinical cardiac abnormalities that may lead to heart failure, including diastolic dysfunction, higher cardiac chamber volumes, and higher myocardial fibrosis. And these will be important to this particular study. We also have some idea what's contributing to this excess risk, and these are just a few examples, but the main point here is that it's a multifactorial issue, ultimately leading to overactivation and dysfunction of immune responses as well as the coagulation system. But our understanding of this is remarkably incomplete, and so we're in need of creative approaches to capture the complex combined exposures and generate hypotheses on some of the underlying biology. So one way we can work to start filling these gaps is using proteomics. Proteins govern so much of what happens in the body, but the epidemiologic literature largely focuses on just a small number of circulating proteins, which ultimately represent just a minuscule proportion of what could be contributing to disease. Proteomics facilitates the larger scale exploration of these proteins that's less dependent on our incomplete knowledge of the biology. And worth noting here is that these tools are becoming increasingly accessible. So using plasma proteomics, we aim to use an innovative sort of exploratory approach to leverage the wealth of information in a larger scale signature to see if we can't use that to agnostically capture some meaningful combined risk profiles. So analytically, that looked like this. So we clustered participants agnostically by the overall similarity of their plasma proteomic signature. Then we described them clinically by comparing various risk factors, including sociodemographics, self-reported substance use, basic clinical factors like BMI and hypertension, HIV-related factors, and then to contextualize to cardiovascular risk, we have cardiac MRI. And then finally, we can use the proteomics to identify some of the biology that are defining the proteome features of each cluster. So the study population was people living with and without HIV, age 40 to 70 years. Between 2015 and 2018, they were co-enrolled in one of 3 HIV cohorts in the United States, Max, Wise, or Alive. There were 352 participants in this study, about 60% of whom are living with HIV. Mean age, about 55 years, a quarter female, 70% Black, relatively high prevalence of substance use that's pretty balanced between people living with and without HIV, including 39% reporting stimulant use in the last 5 years, mostly cocaine, and about a third reporting opioid use. 22% have active Hep C, and then some just traditional CVD risk factors, roughly half have hypertension, half dyslipidemia, and again, fairly well balanced by serostatus. Among people with HIV, 73% are undetectable, median CD4 count about 600, and 88% are on ART and have been on it for quite a long time. So now a few notes on the plasma proteomics that we generated. We used the Olink Explore 3072 platform, and this is essentially just a multiplex immunoassay, but with a more sophisticated probe design and detection approach that confers some nice performance characteristics. It's pretty powerful. And so ultimately, we ended up with about 2,600 proteins measured and analyzable. For clustering, we applied an algorithm known as NMF, which essentially just decomposes the protein data matrix into two separate clusters, one assigning probabilities of cluster membership to participants, and one assigning cluster membership to proteins. And then we take the average over 250 iterations and use that then to assign both participants and clusters to, and proteins to clusters. Using that algorithm, we identified 3 clusters. This matrix on the right depicts mean connectivity or similarity of between measured proteomes. So the higher the value, the more similar the overall proteome. We see these pockets of red here that have high connectivity and are corresponding to cluster membership. Pink, green, and orange we see at the top. So what do these participants in these clusters look like? So what I'm showing you here are what I'm calling distinguishing characteristics, which I define as factors that statistically differ from each of the other 2 groups using multinomial regression. This orange cluster has the lowest prevalence of HIV and Hep C, more socioeconomically advantaged, lowest prevalence of current smoking, and then also have the lowest values of 2 important subclinical myocardial disease markers, left atrial volume and myocardial fibrosis. These markers have differed by serostatus in several studies across world regions, so they are pretty important. This green cluster is sort of the flip side, in some sense, of the orange cluster. Highest prevalence of HIV and Hep C among people with HIV, lowest percent receiving ART, and lowest current and nadir CD4 counts. More socioeconomically disadvantaged, higher proportion reporting opioid and stimulant use, and more adverse values of these subclinical cardiovascular markers. And then the pink cluster is sort of landing somewhere in between, with the only distinguishing characteristic being lowest reported stimulant use. So something I think is important here is that many traditional cardiovascular risk factors are not significantly differing between groups. So I think that suggests that perhaps we're capturing something with the proteome that goes beyond traditional risk factors while still associating strongly with clinically meaningful cardiac phenotypes. So now some of the interesting stuff. So what do the plasma proteomes among these people look like? Among participants in the pink cluster, so kind of falling in the mid-range on risk, we're seeing overrepresentation of processes involved in cellular senescence, turnover, and response to stress, including oxidative stress, cytokine-induced NF-kappa B activation, these stress-induced cell proliferation pathways, and I'll point to mTOR signaling, which is an important growth regulator that coordinates cell growth and metabolism with environmental cues. This green cluster, so kind of higher cardiovascular risk, we see more of an immune activation signature, higher levels of proteins involved in IL-10 signaling, which is an anti-inflammatory cytokine, and IL-12 signaling, a cytokine involved in differentiation of T helper cells and cytotoxic activity of CD8 cells and natural killer cells. We also see enrichment or higher interferon gamma production and activation and migration of macrophages, B cells, and T cells, including a handful of specific subsets there. Lastly, some processes involved in tissue homeostasis and turnover, including insulin-like growth factor signaling, Notch signaling, and the MAP kinase cascade, as well as angiogenesis. So then lastly, this sort of lower risk group, we're seeing lower plasma abundances of proteins involved in pro-inflammatory cytokine signaling, interferon signaling, B cell and platelet activation, and then lower markers of cell senescence and response to stress. So a summary, that was a lot, so I'm going to try to kind of simplify it here. We've identified 3 clusters of people living with and without HIV based on their plasma proteomic signatures. This pink cluster of 91 participants has mid-range, or I'm sorry, the orange cluster, we'll start with the orange cluster, lowest HIV-related cardiovascular risk based on subclinical cardiac abnormalities, lowest prevalence of HIV and Hep C, more socioeconomically advantaged, and proteomic signatures reflecting lower activation of tissue homeostatic and immune responses. This pink cluster, representing sort of mid-range cardiovascular risk, have proteomic signatures reflecting upregulation of classic tissue homeostasis and cellular senescence pathways. And then this green cluster, sort of the highest CV risk, have people living with HIV with lower ART use and CD4 counts, highest prevalence of Hep C infection, higher substance use, more socioeconomically disadvantaged. And then proteomic signatures reflecting upregulation of T cell, B cell, and macrophage activation, as well as cell metabolism and turnover pathways. So just a couple final points here. We used a sort of unique and exploratory approach to identify subpopulations of people with and without HIV that may reflect systemic biology underlying interactions of exposures to risk factors for both HIV and cardiovascular disease, which are manifest by subclinical abnormalities in cardiac structure. Which is kind of, and the results are sort of implying to us that the proteome may objectively capture some complex combined exposures beyond traditional cardiovascular risk factors that could potentially be leveraged toward improved risk prediction and targeted interventions. So with that, I will say thank you to all my co-authors, funding sources, and of course, all the study participants. Thank you.
Yeah.